I direct a neuro-engineering group at the MIT Media Lab, and our goal is to try to reveal new inventions that allow us to make progress in understanding and engineering and repairing the circuits of the brain. Now, the last century of neuroscience has taught us that the brain is an incredibly complex circuit made out of hundreds of billions of cells called neurons, and this is evidenced through all sorts of studies, animal model studies, studies of human patients. These neurons, of course, are incredible computing machines. Each of these cells is connected to some tens of thousands of others, perhaps, and they compute using millisecond timescale electrical pulses. But it's also the case that these neurons have all sorts of modalities of communication and computation that we're just learning about. For example, neurons are known to release gaseous messengers and allow communication across volumes, for example, not just through their connections. And so we're kind of in this interesting era where we're trying to build technologies to reveal fundamental principles of brain operation, while we're also trying to figure out how to use these principles in order to repair brain circuitry, and even, as I'll show you at the end, to try to build brains from scratch in a dish. One thing we know about the brain is that it's incredibly dense and heterogeneous. So within a cubic millimeter of brain tissue, you have perhaps 100,000 cells connected by something like 1 billion connections. Each of those connections can also be active at the millisecond timescale. So if you think about it, that means that that cubic millimeter of tissue could, in theory, be capable of 1 trillion different distinct events per second, although it probably has even more subtle things going on within the cells. But it's not even known how many kinds of cells there are in the brain. This is sort of how little we know about what's going on inside of our heads. We know that there are some cell classes that are distinct. For example, these small star-shaped cells, known as basket cells, are amongst the cells that are compromised in patients with diseases like schizophrenia. And we also know a lot about long-range excitatory projection neurons that will send information throughout even large fractions of the brain. And these are amongst the cells that might be overactive in patients who have epilepsy, for example. So we need to figure out how to build technologies that allow us to answer fundamental questions, like how many kinds of cells there are, how are they connected, what molecules are at those connections. And also, how do we repair these brain circuits, both at the anatomical and molecular level? Can we actually form connections and even build parts of the brain if we need to? But also at the dynamical level, can we enter information into the brain in order to resculpt the computations that are happening? So what we're trying to do today is to talk about technologies that we're building in order to try to enable this kind of approach to happen. We build tools that we then disseminate to the community, and over 1,000 groups around the world are using these tools from our group to analyze, engineer, perturb, and construct brain circuits. But our main goal is to try and figure out how to push the technology in new directions. Now, of course, this is important not only for philosophical reasons. We want to understand how the brain computes, but also, as you've heard, to address fundamental medical needs about the brain. The brain, of course, is not only important for our lifespan and our ability to move and other things like that, but also it obviously is highly important in generating our identity and our feelings and our consciousness. And so that's why the fact that these disorders of the brain, pretty much none of which can be completely cured and very few of which can be treated, are so devastating. Now, of course, there's been some success, and you've heard a little bit about neuroprosthetics using electricity, for example, to control brain circuits. And of course, pharmaceuticals have had impact over the last 100 years in treating brain disorders. But one could argue that even a well-trodden field like brain pharmaceuticals is an area in need of great innovation. So these are some numbers from an article in Science magazine a couple years ago, where they point out that brain drugs currently take about 9 years to be developed and approved. They fail about 92% of the time to get clinical approval, and the cost per drug is enormous, about $850 million each. So if we think about it, a drug that just bathes the brain in a chemical might affect circuits in the brain that you want to fix, as well as circuits in the brain that you'd rather leave untouched, and that, if the drug does perturb, will result in side effects. And so part of the goal, I think, is not only to think of new prosthetics and ways of controlling brain circuits, but even when we are thinking about pharmaceuticals, are there ways for us to figure out how to make them target circuits in the brain? Now, I want to talk about a couple different projects that we're working on. This first one is a project that we're working on with Craig Forrest's group at Georgia Tech, which is to try to find a way to analyze the cells of the brain. As I mentioned, we don't even have a complete list of the cell types of the brain, a parts list, if you will, of the basic circuit building blocks. Now, the brain doesn't feel pain, so we can actually, in the living brain, introduce robots into the brain that can then help us figure out what are the integrative properties of cells in the brain. If we can record the electrical pulses in a cell, and we can also observe the molecular contents and look at the morphology, that would tell us something important about how that cell works. So what you see here is a robot that we've built where we introduce a very small glass microneedle into the brain. And what we're trying to do is to detect a cell and to stop when we get to that cell so we can do automated neural recordings, automated surveillance of the molecular contents of cells, and also injection of dyes into the cell so that we can see the shape. And the way we do this is we deliver little electrical pulses down this microneedle so that we can sort of do an impedance survey of the environment. The closer we get to the cell, the less current will flow through the pipette. You can think of it as sort of getting blockaded by its environment. By doing a time series analysis of the electrical impedance of this needle tip over time, we can actually predict when we're going to hit a cell with high accuracy, perhaps 80% nowadays. Then, once we've detected a cell, we can apply a bit of suction. This is called the patch clamp method, which we did not invent, but we have been able to automate it for use in the living brain. And when we apply suction, we can now have an intimate interface between our microneedle and the cell. And that's exactly what we want to do if we want to get intricate and integrated surveillance of what's going on inside the cell. So if we apply a little bit more suction, we can actually get access to the inside, and now we have an electrical connection to the inside of the cell. So that, for example, if that cell fires an action potential, one of its electrical pulses, we can observe it. We have an amplifier that's connected to this microneedle. Similarly, we can inject a dye into the cell to see its shape, and of course, we can now harvest the contents by sucking it out. So now we're working on a variety of other strategies. Can we scale up these robotic arrays to be able to do the analysis of many cells in a circuit? Can we do this in the awake brain, for example? So this could even be used as a neurosurgical tool to give synapse-level information about what's happening in cells in the brain to help people who are undergoing neurosurgery for various conditions. By being integrative, we can connect the dots. We can associate electrical, circuit, and molecular properties all in the same cell. And this is something we're now working on as we think about how to make nanorobotic strategies for doing this as well, so this can scale up to significant fractions of the brain. Now, these are destructive to the cell. If we harvest the contents, the cell no longer has its recipe to make it do what it does. So we also have been working on extracellular methods to tile the brain with electrodes, so we can record cells in circuits distributed throughout an entire brain. What you see here are some microfabricated devices we're building with Cliff Fonstad's group at MIT. And at the top, you can see a three-dimensional array that has prongs that can span a significant fraction of, let's say, a mammalian brain, such as a mouse model that's used commonly in neuroscience. If you zoom in, as you see on the bottom here, we're packing electrodes incredibly dense in the sense that they are sort of tiling these electrodes. So in some ways, this is almost like an imager of the brain. We're imaging the electrical activity by picking it up in a 3D pattern throughout an entire brain circuit. And that's a very powerful way to collect information about the brain, because if you can pick up neural activity in regions that are mediating sensations, emotions, decisions, and actions, you might be able to connect the dots to explain how these different cells are working together to integrate information towards goals or decisions or complex behaviors. Now, we also want to be able to fix the brain. And to do that, we have to take a step back and think about how neurons compute. Now, of course, as we talked about earlier, there are all sorts of messengers, even gaseous ones, and different ions and kinases and all sorts of things that mediate neural computations. But a very useful abstraction has been to think about neurons computing using electrical pulses. And so if neurons are computing using electrical pulses, and if we were able to find essentially the molecular equivalent of solar panels that convert light into electricity so that we can install them into neurons, then shining light on those cells should allow us to turn those neurons on or off, depending upon the way that those electrical charges move. And as you mentioned, the brain doesn't feel pain, so we can implant prosthetics into the brain, including light-emitting prosthetics, like an optical fiber that you can put into the brain. And now you can deliver light to a circuit to turn it on or off. So over the last 40 years, many groups have been able to find molecules that will convert light into electrical energy. And a couple years ago, the Hegemon, Nagel, and Bomberg groups working together in Germany were actually able to pinpoint a molecule that's used in unicellular green algae to help it swim towards light. And this molecule is found in a little eye spot in the back of the algae that helps the flagella turn in an optimal fashion. Now, if you zoom in on the eye spot, you actually can see these little proteins respond to light, because if you were to shine light on them and record the electrical currents, you can actually observe electrical currents moving through these little pores. So if you think about it, that's exactly what we want. A molecule that when we shine light on it will respond by changing the electrical gradient across a membrane. Now, we were lucky. It turns out that this molecule is a protein, and it's encoded for by a single gene, and that gene is small enough that you can actually put it into other cells using techniques from the field of gene therapy. So we can take the snippet of DNA from this algae and put it into a vector and deliver it to neurons. This is a collaboration that Nagel, Bomberg, Dijsselroth, and I did in 2004 or so. Now, this is where serendipity kicked in. In the algae, this molecule is expressed in a small eye spot inside the cell. But when neurons received this gene, interestingly enough, they were able to manufacture the protein and install the proteins on the boundary or the membrane of the cell. So that was one piece of luck. A second piece of luck was that the currents that were mediated by this protein were just the right time scale and amplitude that when we shine light on a neuron, we could turn it on. It would be able to mediate those fast electrical pulses. So I always tell students when they start out, the most important thing is to be very lucky when you're doing a project. So this is great because now we can actually aim light at a cell, and unlike electricity, which will go in all directions, activating not just neurons we care about, but all sorts of other cells that might be just the kinds of cells that we don't want to affect, we can now manipulate neuroactivity very precisely. Okay, so how does this work in practice? Well, let's take that basket cell, the one that's compromised in patients with schizophrenia that we talked about earlier. It turns out you can take a little piece of regulatory DNA, put it in front of your light-activated protein, or opsin as they're known, and then when you deliver it to the brain, only these cells will have the factors that turn on this piece of regulatory DNA and make the molecule. So you can actually target just these cells and not their neighbors, even though they're all densely packed together, so that only these neurons are light activated. And now when you bathe this circuit in light, these cells will be active, but their neighbors will not be directly affected by light. So imagine that we could take cells that atrophy, and if we can augment their performance in some way, maybe we could repair computations directly. Or for many cells of the brain, we actually just don't know what those cell types necessarily do. Or if we have a hypothesis, it's hard to prove that those cells do something specific. So I'll tell you about one concrete example here. That's a basic science point, but a useful one, which is to try to understand which brain cells drive the processes in the brain that mediate sensations like reward or pleasure or addiction. And I think a lot of us have heard of dopamine, of course, and I think in the media it's often colloquially referred to as a pleasure center of the brain. But it's an open question of what exact patterns in these neurons will drive a sensation that will reinforce what whatever the brain was just doing. So we collaborated with the Fiorilla group at KAIST in Korea, and we're able to label through these acronyms at the top just this set of cells, the dopamine-producing cells deep in the brainstem, a tiny cluster. And we were able to put the molecule from algae, called channelrhodopsin 2, just into those neurons. So when we shine blue light on them, they'll be activated. The experiment then was to do a very simple behavior. To take an animal model like a mouse, whose neurons that make dopamine have been made light sensitive, and to shine light on those neurons whenever the mouse goes to a certain point in this box. So when the mouse goes to the blue dot, it'll get a pulse of blue light directly down an optical fiber that goes into its brain. And there's a control point, that black dot, where if the animal goes there, nothing will happen. So this is what happens. The mouse is going to this little right-hand portal, and every time it pokes its nose into a little sensor, it gets a pulse of blue light delivered into its brainstem. And as you can see, the mouse pokes its nose, gets a pulse of light, and what this pulse of light's doing is activating those cells and making the brain do more of whatever the brain was just doing. And the mouse doesn't go to the other site, which has no associated rewarding contingency. So this allows you to prove that 1/5 of a second duration light pulses are enough, just one pulse at a time, are enough to make the brain do more of what it was just doing. And that's very important if you want to understand the basis of sensations like reward or pleasure, but also things like compulsions, right? Or situations like addiction, where these circuits might be hijacked. Now, in the years since, we've been branching out and looking all over the tree of life for molecules that can do this. And it turns out that pretty much every kingdom of life, except for animals, have some molecule that will convert light into direct electrical gradients. And so, for example, we've now collaborating with this 1,000 Plant project that's headed by Gain Wang and involves folks like Michael Malkonian and others. We've been able to look at literally thousands of new molecules that convert light into various kinds of signals. And so one could argue that analogous to synthetic biology, we're sort of seeing a synthetic physiology era, where instead of just controlling genes, you can now control real-time dynamic processes in living cells. And we've devised all sorts of high-throughput optical equipment to screen for these molecules, because they're sometimes very hard to find. Let's describe one of these molecules that is of great use and is now in use by hundreds of groups around the world, which is sort of the opposite of the molecule we looked at before. The channelrhodopsin 2 molecule I told you about earlier, when you shine light on it, brings positive charge into the cell. And several years ago, we found that a molecule that puts negative charge into a cell can also be put into neurons and successfully expressed. When you shine light on neurons containing this molecule, it will now allow those neurons to be shut down with light. So you can imagine doing all sorts of different things with this molecule. For example, could you turn off neurons that are involved with epilepsy to shut down a seizure right when it occurs? And actually, many groups are now engaged with such research activities. Here's an example from Akihiro Yamanaka's group, where they again labeled a tiny cluster of cells, but this time they made them silenceable with light, not activatable by light. And they labeled the sets of cells in the hypothalamus that are known as the hypercretin or orexin cells, and these are cells that are compromised in patients with narcolepsy. So the question still remains, if these cells are gone in a patient with narcolepsy, does that directly lead to a sleepy state? Or is the loss of these cells going to compromise other brain circuits that then adapt over periods of months or years, and those losses are what cause the chronic sleepiness and other changes that characterize narcolepsy? So what Akihiro's group did was to make a transgenic animal with just this small cluster of cells deep in the brain made sensitive to light. And then they put a laser connected to a fiber, this time a yellow laser, so that you could turn these neurons off. And what you can see here is this is the probability of being awake over time. When they turn the yellow light on, the animals fall asleep within some tens of seconds. And then when you turn the light off, they wake back up. Although if you look carefully, you can see they're a little bit groggy still near the end there. So this allows you to prove that these cells, once they're deactivated, can result instantaneously in a sleep condition. Now, with exquisite control over cells, and you've already heard about some of the things that can be done with electrical stimulation, but now we can turn on or off specific cells embedded within the dense matrix of the brain. One very exciting area is to think about potential clinical applications. And, you know, that might seem a bit far out in the beginning, but if you think about it, over 250,000 people have some kind of neural implant already, cochlear implants, deep brain stimulators for Parkinson's disease. We have a molecule that has to be genetically expressed in neurons, but gene therapies are also seeing, thanks to new viruses like the adeno-associated virus, a resurgence of interest. And in fact, a gene therapy was recently approved in Europe. And recently, we've collaborated with groups like Bob Desdemona and Anne Graybiel to do non-human primate testing of these molecules to see how they are tolerated by the brain. Because, of course, these molecules come from plants and bacteria and so on. And so we want to know that the immune system or that cells won't regard these as foreign. And so the initial data is a small data set, but more groups are now replicating them. And so we're very excited to see how this avenue of preclinical testing goes in the years to come. Let's consider one concrete application area, which is in the field of blindness. So the retina in the back of our eye contains photoreceptors that capture light and transform those light signals into signals that can, through a layer of neurons, be transmitted to the brain. And many patients have lost the photoreceptors, perhaps millions around the world, and can no longer do that initial light capture step. Here we are zooming in into the retina in the back of the eye, and those patients who've lost their photoreceptors will be lacking an entire row of cells. But there are still spared cells in the eye, and furthermore, light can still get into the eye. So what if we could take the molecules that convert light into electrical energy and install them in one or another layer of the cells that are spared? Maybe we can convert the rest of the eye into a high-resolution camera. So this is something that we've been pursuing in projects led by Alan Horsager and also being explored by a startup that I'm involved with called EOS Neuroscience. Here's a blind mouse. It's trying to solve a maze. In this maze, there are 6 arms. And there's some water in the maze because mice like to swim, but they like to sit on platforms more. And so this mouse is swimming around, and it's trying to get to a platform that's at the top of this image. Now, this mouse is blind. It turns out that many of the mice that you find, for example, at pet stores, have mutations that cause photoreceptors to be degenerate. And so mice are smart. They can solve the maze by systematically searching each of these avenues until the target is found, but the mouse is not using vision to do the task. The acronyms refer to different mutations that more or less address different kinds of human blindness that the field of ophthalmology has discovered over the years. Now, this mouse was blind, say, a month or 2 before, and received 1 dose of an adeno-associated viral vector, and that contains the channelrapsin 2 gene. And what you can see now is that mice can very quickly navigate to the platform. In fact, they can navigate as fast as mice that have seen their entire lives. Now, it doesn't prove that they're having, say, conscious vision and seeing form and shape. But it does show that they can make cognitive use of their visual information in a way that they were not able to before. In this study, we also showed that the eye did not have responses that are indicative of an immune attack. And so it's all in mouse, but now we were collaborating with many groups to try and see how this might apply either to different cells within the retina or potentially in different animal models that might be more accurate than the mouse, say, maybe something like a primate model. Now, the eye, of course, can be accessed from the outside. But what about the brain itself? It's very dense, it's huge. How do you get light into arbitrary 3D patterns in the brain? So with Cliff's group, where we worked on this 3D electrode recording device that I showed you earlier, we've also been working on 3D light delivery devices. And the way these work is that they're essentially microfabricated optical fibers that are all packed together. So what you see here, each blue line is essentially a tiny optical fiber, perhaps 10 microns wide, and they conduct light down this probe until you get to the desired depth in the brain, where then a mirror will steer the light out at a 90-degree angle. And recently, we also developed ways to scale this up into three-dimensional devices. So now we can play back light into a 3D pattern. Now, there aren't any light sources shown here. What we do is we hook it up to a projector, not unlike perhaps the kind that are at the back of this room. And now the projector is essentially a megapixel light source. You can play back a 2D pattern, and our array will then convert that 2D pattern into a 3D pattern. So we can play back arbitrary patterns to the brain. Or, for example, spell out MIT, which I'm not sure what the brain makes of that, but it's kind of something MIT people do. So recently, we started to implant these devices into the living brain. These are, again, using mouse models, as is commonly done. And what we can do is to play back sort of arbitrary patterns into the brain. So we can now dial in sequences or drive synchronous patterns or try to engage one part of a circuit and then another. And so one hope is that with these technologies, we can start to sort of speak more naturalistic neural codes with the right spatiotemporal resolution that the brain expects. So for example, we can actually play back a pattern in one part of the brain while we record neurons in another part of the brain and try to figure out, is there an optimal pattern of control for one circuit to make another part of the circuit do something? And so this could be very useful in terms of sort of screening for neural codes that might be therapeutic. Now, there's obvious implications for sensory prosthetics. In this case, we actually are implanting this into the sensory cortex. But there's also a lot of potential areas where you can make cognitive prosthetics of sorts. And so this is an area we started to think very hard about. Could you build essentially brain coprocessors, devices that can read information from the brain, using, for example, those very dense 3D arrays that I showed you earlier, then, through custom computers that we're designing, compute exactly the information that's needed in the brain, and then finally, to use three-dimensional, high temporal precision, high spatial precision devices to enter information back into the brain. So this can be very useful as a general architecture for prosthetics. And one of the ideas is that, in sort of neurodegenerative conditions where large fractions of the brain might be compromised, could you actually try to build replacement parts for the brain in the style of a coprocessor that might work with a computer chip? It's very interesting to think about computers. We are using currently computers that are implemented in the same way that everybody else does it, in silicon. And one of the things that we're thinking a lot about, though, is if neurons have such incredible information processing capacity, and also a genome that allows it to express different kinds of functions at different times, and all sorts of molecular signaling cascades that can integrate information over very different timescales, what if we built computers out of neurons themselves? In the short term, this could be very useful, because now you could actually build neuron-based computers in a dish and use that to explore, in a constructive style, how to assemble neural circuits. What are the rules through which different cell types wire up? And how do you understand how these circuits that you build from scratch compute? Richard Feynman once said, "What I cannot build, I cannot understand." And one of the ideas is that neuroscience could benefit from constructive approaches where you try to build brains in all of their glory by really trying to test our theories of how cells wire up and how they form connections and activity. In the long term, of course, should this line of research prove valuable, it might actually be useful for building replacement parts for the brain that have realistic cell types, connections, and so on, and have properties of the brain that are hard to mimic, including all of the computational properties of neurons, but also the very low power usage of neurons and the ability to intimately wire up with existing neural circuitry. So we've been collaborating with the tissue engineering group of Utkan Demirci to explore this. And what we're doing is using photolithography, the same method you use to make computer chips, to try to deposit cells of defined kinds at defined points in 3D space. Because unlike other forms of tissue engineering, the brain demands an incredibly three-dimensional, high-resolution method of fabrication. So essentially, we wash in a monomer in a solution along with some neural precursors. And then we shine light through a mask, or you can do it with a projector as well, and then aim light so that only some parts will receive light and others won't. And wherever light is hit, we'll cross-link those monomers into a solid and trap whatever neural precursors were there at that point. And then we can wash away all of the other monomers and cells and repeat this. So very rapidly, you can start to build up little neural circuits. For example, you can, as you can see here, build a little cube of this polymer and trap a cell at a defined point, and then build sort of a little highway path so that there are regions where the axons and dendrites can wire up. And one of the goals is to build this in a scalable fashion. Can you do this at brain scale, which will probably require us to also think about how to integrate blood vessels and vasculature into the mix as well. I wanted to close on a note about how we're trying to build tools that are powerful, but that we can rapidly deploy in the community. We try to give away tools however possible to accelerate research. But also recently, along with Joost Bonsen, we started a class called Neurotechnology Ventures, and I've been trying to figure out how to get people to get their ideas out of the lab and into the world at scale as well. And part of my hope is that through these efforts, we can build the neurotechnology discipline, but also help contribute to the neurotechnology industry as well. So this is a vast collaborative effort. There's not time to name everybody here, but I'll close on this slide, and I thank you for your time.